Assalamu alaikum. I hope you're all good. Today I'm going to explain more about the structural details of a protein. We have already discussed primary and secondary structure in previous lectures. So let's do a quick recap and let's get kickstart. Here we'll be revising the topic of protein structure. As we know, protein structure is made up of four components. Protein exists in four different forms for different types of structure, as we already know. So here we have the primary structure and we have the secondary structure, we have the tertiary structure, we have the quaternary structure. We are already done with primary structure and the secondary structure. So let's do a little bit revise of it. In primary structure, what have we found is primary structure is just the sequence of amino acids. Amino acids are aligned, they are joined together in a sequence, in a line arrangement, and which is guided by the mRNA, and this sequence is made by ribosome, the protein synthesizing factory. In the left hand side or the left end of it, we have the first amino acid. Once these amino acids are coming out of the ribosome, we have the first amino acid, then the second, then the third. This end is known as the first end, and this end, the first amino acid in it, has the end terminus, amino terminus on this side. So this first amino acid is also known as end terminal amino acid, while the right end, after the completion of whole protein structure, after the whole primary structure has been formed, we got a long strand of a protein, we got a long strand of polypeptide. We have two ends. This is the left end, which we call N end, and the last amino acid at this side, it has the free end, which is the carboxyl, and we call this as C terminus or carboxy terminus. Now, what is a secondary structure? In secondary structure, as we have already known, protein is not just a linear, as we have seen here. It's just not like this a linear structure, but amino acids, rather being in a linear structure, they join together, they just do the internal bonding because of hydrogen bonding, hydrophobic interaction, many other interactions are involved. So a protein assumes a secondary structure. The first one is the beta sheet type of structure, in which proteins are arranged in a sheet like manner, and the second one is presence of a structure which we call helices or alpha helix. This regular repeating local structure give secondary structure to a protein. These are just the secondary structure. The first one example we can quote here is fibrous protein, like we have keratin in our nails, in our hair, hoofs, claws, actin, myosin protein. These are the proteins which are present in muscle cells, which are involved in contraction and relaxation. Collagen also a protein which is a fibrous protein, and it exists in secondary structure. Now here we are going to discuss the tertiary structure. What is the tertiary structure? How is it known to us, and what do we need to learn about it? Tertiary structure, this is just the highly folded part of a protein. In this structure, we have a 3D globular structure of a protein. It's actually overall shape of a protein, a spatial arrangement or relationship of secondary structure. As we have already seen, there are the beta sheet type of structure and we have the helix type of structure. These structures actually interact with each other so as to result in a tertiary structure. Relationship of secondary structure to one another give us tertiary structure. As we can see here in this short video, we can see this is actually the tertiary structure formed by actually combination of different secondary structures. As we can see here, there are two types of secondary structure. One is the alpha helix type and second the sheet type. So these combine together to form a tertiary structure. Actually, the interacting groups are the R groups of amino acids. Different amino acids have the R groups. These R groups are actually combining together. They are actually making different types of bonds which help it to make a tertiary structure. Now, what type of bonding do we find in a tertiary structure? There are a lot of different types of bondings. We can find them in a tertiary structure. So we can have a salt bridge, we can have ionic interactions, we can have disulfide bond, we can have hydrophobic interactions, van der Waals, London force, dispersion force, hydrogen bonding, a lot of them. We don't need to discuss them here. We just need to remember there are a lot of interactions going on between the R groups of different amino acids. That's it. Now, what about the quaternary structure? What is it? Actually, this quaternary structure is just not one polypeptide. As we have already seen, this is a single polypeptide, a single chain of amino acid. Actually, what is a quaternary structure? We have two or more than two polypeptide chains of these amino acids. Okay, they just combine and make a single functional unit like this. As we can see, there are different types of colors. We can find different types of color which are just ribbons, which are just locating and just moving around. They depict different types of polypeptides which have been joined together by different types of interaction. Again, every protein which we know till now does not have the quaternary structure. It means every protein is not made up of more than one polypeptide chain. Okay, it's not necessary that a protein should be made up of more than one polypeptide chain. That means some proteins are just made up of just one polypeptide. Okay, the same type of bonding as we have already discussed in the tertiary structure, same type of bonding is present here also, which we already know. Okay, where both interchain and intrachain, these are the two important terms to understand. Interchain hydrogen bonding is between two chains, and see, this is the one polypeptide chain and this is another polypeptide chain. Once there is an interaction between two different types of chain, we call that interchain bonding. And intrachain means this is just a single chain. This contains the N and C terminus, and this in the same chain of polypeptide we find these bondings. So this is the explanation, simple. And we can see the example are hemoglobin, antibodies, insulin. These are the proteins, these are different types of proteins which exist as a quaternary structure. Here we leave it. This is the end of our lecture of protein structure in nutshell. Thank you so much for joining me. If you enjoyed the video, please hit the like button, share this video with other students, subscribe to my channel, press the bell icon for sure. Thank you so much guys.